Share screen. Okay. So good morning to everyone. Today we are going to report our research in biotechnology. So here are my main members and teammates of this group report. First is the leader of this group is me, Mr. John Joshua Di Calisanga. Second member is Mr. Aljon Kuhay. And third member is Miss Maria Loida Jordan T. So let's proceed, shall we? Okay, so here are the main objectives of biotechnology. After this presentation, you will be able to know the meaning of biotechnology, history of biotechnology, types of biotechnology, this of biotechnology, importance of biotechnology, pros and cons of biotechnology. So, history of biotechnology. In 1919, a Hungarian agricultural engineer, Karl Erecki, foresaw a time when biology could be used for turning raw materials into useful product. Um, it it was Karl Erecki is the first person who could discover that it is truly it is truly helpful for humans to use raw materials into useful products. Even in today's generation, raw material products are still being used. Even recycled products, let's say natural resources, can keep the harness of creating such products. Like for example, native products. Here in the Philippines, we have a lot of native products uh, like native bags, native sleepers, native baskets, native wallets. Those those products are made out of woods, bamboo, or even the skin of those woods, or like that. For in fact, the biology term was studied by Carl Erecki. Winnie found out that it is very helpful that raw materials can be used into useful products because um, even though even though natural resources are still very efficient at that time when producing products that we use today, like papers. Paper is made out of leaves or silk. uh, Clothes is made out of silk. That's it. The breakthrough that laid the groundwork for modern biotechnology came when the structure of DNA was discovered in the early 1950s. So at this time, it was the era where DNA was first discovered in the early 1950s, where it became a source for manufacturing, testing results, and uh, bio biotechnological processing for groundworks. Like when we say biotechnology, it is structuring something to produce technological processes that will be discussed later on this next slide. And that groundwork was first discovered in the 1950s. One of the main source of DNA is very useful to scientists because it helps them to clarify the results of background study of plants, humans, or animals. But in that year of 1950s, it was how DNA was invented. It is when DNA has to be used for sample tests like that. Okay, so let's proceed. What is biotechnology? Biotechnology is technology that utilizes biological systems, living organisms, or parts of this to develop or create different products. Many forms of modern biotechnology rely on DNA technology, and it goes through biological process such as fermentation and harness. biocatalysts to become manufacturing plants. So in the early years of 1950, DNA was structured and first discovered and invented, right? So back in the day, people use sample tests of plants to be manufactured and to be produced. So they do testing of a plant background and what is the growth or the origin of that plant, like a seed or like flowers or like plants that can even it's not a seed but they still put tests and put some put some experimentation towards it in order to rely on its biological effect, or how does this plant grow, when does this plant grow, how many years or months does this plant would be able to evolve like trees. Okay, so trees can evolve in years, right? It even there are 100 years old tree or even like that. And that is how DNA technology goes in processing of plants, manufacturing plants. So in today we have product plants that is packed, so which are seeds, mostly are seeds. So they use it for manufacturing. But before that, they test it into a DNA to test the plant's background and its historical uh, origin. So here are the main two topic of biotechnology processing. First is fermentation. It is a metabolic process that produces chemical changes in organic substrates through the action of enzymes. Fermentation is processing that produces chemical change, which the dynamic resolution of change can turn into uh, something different. Like for example, if you put water and then you put food coloring, they dissolve with each other, right? You will see the substrate that the action will spin around because the water dissolves with the chemical reaction of the food coloring out there. So it changes the dynamic of uh, the chemicals between that bonds together, which we use to be termed in chemistry. Or like for example, you put you put oil and water together. They don't collaborate, but they differentiate because water is pure liquid, while oil or cooking oil, let me say, cooking oil is a sticky liquid. So once you put the oil, it does not sink down under the water. It just stays above. It does not change, but it changed, but it does not collaborate with the chemical reaction. But the only change was there is the water does not react while the cooking oil didn't react also, because the sticky liquid is firm while the pure liquid is so like smooth, but also uh, what we call that, very a harnessing liquid to be exact. Next is DNA technology. DNA technology is a sequencing, analysis, and cutting and pasting of DNA. So to sequence the analysis and cutting pasting of DNA is how technology revolves around DNA tests and samples. Like how do we get results from biological terms if DNA technology produces a lot of technological helpful structures, like not only for plants or animals background, but also in humans also. Okay, sorry. Okay. Also in humans that takes to work on to find out who is the biological parents of this child, or in today's generation, even before, uh, DNA is very useful for crime investigating, or crime investigators use sample of DNA tests to 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 to, to research or study a person's if a person's criminal background, a person's victim's background, the blood sample test or fingerprints. DNA is also included there, as well as it it helps them to track or trace something. That's why DNA technology is very, very important. Okay, and that is what called analyzing and sequencing. Um, who are the father of biotechnology? So here we have two main fathers of biotechnology. First is Carlo Erecki. German Karl Erecki is born on 20th of October 1878 and died on 17th of June 1952. He was a Hungarian agricultural engineer. He is regarded by some the father of biotechnology because the term biotechnology was coined by him in 1919. So Carl Erecki was the one who found out that in biology we can use raw materials into useful product. Meanwhile, it was him who termed the name biotechnology because of his study that there is a change in processing by the use of technology. But even at this time, technology is only limited. He still discovers the changes in producing goods, materials, and products by the use of technology that helps evolve throughout the years. It was during his time, so he is regarded by the father of biotechnology and was coined by him in 1919. Second is Louis Pasteur, 1822 to 1895. The French chemist and microbiologist is considered the actual father of biotechnology. By most renowned scientists of the world, it is he who had established the role of microorganisms in the fermentation process for the very first time in 19th century. So Louis Pasteur is a chemist, microbiologist, who had established the role of microorganisms or little particles, little organisms, because we say micro. He established and studied his experimentation throughout for the very first time that it was him. At the time, it was him who discovered microorganisms into uh, living, living, living life or living organisms. And because of that, he is renowned. He was by most renowned scientists of the world because of his role and had established the study of microorganisms. Further with the statement, his study was kept to be an evidence that microorganisms is also a living organism, which is part of the microbiology study. So as I say, he is the actual father of uh, biotechnology because of his experimentation and uh, establishing of the role of microorganisms, which are the little living organisms. That's what I say. So that's all for today, guys. Thank you for listening and hope you will learn something in our report.